So my great pleasure to join you for this seminar. All right. So this is the outline for today's talk. First, I will spend about 10 minutes or so to talk about introduction and the background for the 3D genome structure. Then I will briefly introduce the current method and the technologies and how we use to study 3D genome. And then, well, the lecture will move on to the relationship between 3D genome and gene regulation. And finally, I will introduce you some tools and resources for you to explore 3D genome data and how you can use them for your own research. Okay, so again, so this is a lecture rather than a conference presentation. I really hope if you have any questions, you can stop me anytime and we can turn this into more like a roundtable discussion rather than I tell you everything I know about the 3D genome. All right. So I will start my lecture with this quote, hope it's famous quote. Life is like a bowl of noodles, right? Not a box of chocolate, right? So in this case, what do we mean by noodles? Of course, noodles are DNA, and the bowl here is the nuclear envelope. You can see different colors. The noodles means there are different chromosomes. So you can tell that different chromosomes have their unique space inside cell nucleus. So these are so-called chromosomal territories. Let me change it to laser pointer. Fantastic. All right. And then when you further zoom in, you can begin to see the human genome is organized into different compartments, something that we are really familiar with, the euchromatin and the heterochromatin. So inside of each different compartment, there's some kind of the hairball-like structure. So we will spend a lot of today's lecture on those hairballs, in which we term TADs, or topologically associated domains. And when you further zoom in, this is the highest resolution. You begin to see the chromatin looping events that we are more familiar with. So chromatin looping is more directly related with the gene regulation, as I'm going to demonstrate in the next slide. Okay. So this is really just the basic diagram for gene regulation in the mammalian genomes, right? So in the center, we have a promoter sequence whose role is to recruit RNA polymerase and initiate gene transcription. So far away from the gene promoter, there's an element called the enhancers, whose main role is to recruit tissue-specific or developmental stage-specific TFs. Right? So I believe the enhancer concept has been covered in your previous lectures. For example, I'm sure you have learned how to use ChIP-seq for his modifications, or use ATAC-seq to identify enhancers in the human genome, right? So although enhancers can be located far away from the gene they regulate, so they do play a critical role in gene regulation. This is one of the most famous example. In this case, this enhancer is located more than 1 million base pair away from the gene they regulate. And actually this enhancer is located inside intron of another gene, right? But when the scientists do experiments, when they delete this distal enhancer, there's a severe phenotype, right? So there are two mice. You can tell which one is good, which one is bad luck, right? So in this case, the guy on the left is wild type, seems very well-fed, a little bit chubby, right? But still looks very furry and cute. The guy on the right, this unfortunately is a mutant. So the enhancer sequence is deleted and no exon is affected. But still you can see the limb for the mouse is severely underdeveloped. So there are quite a few concrete examples like this to show the effect of enhancer manipulations, right? Again, I think these slides probably have been covered by Shirley before, but I just want to talk about it a little bit more to make sure we're on the same page, right? So far, there has been thousands of GWAS studies has performed in the human genome. For example, there might be more than 5,000 or 6,000 SNPs. They are related with different human disease, such as cardiovascular disease, or even cancer, or different human traits, right? But one of the surprising findings from the GWAS study is actually over 93% of these these SNPs, this disease or trait-related SNPs, they are located in the non-coding part of the genome. Okay, and then another study showed that actually majority of these GWAS SNPs, they are located in DHS sites, which means potentially means open chromatin regions in the genome, right? And then the idea is actually if there's some mutation happened in the distal SNPs through Chromatin looping, these defect enhancers might contribute to the misregulation of the target gene through Chromatin looping events, right? More recently, people began to show that even without mutation on the enhancer itself, right, when you only disrupt the 3D genome structure, you begin to severe phenotype in the human body. In this case, this is in the wild type. There are two Chromatin domains. In this case, the domain 1, they have this green band enhancer sequence. And in the domain 2, there's oncogene. So normally, because each element have their unique space, so enhancers don't really talk with the oncogene. But when the patient have the IDH mutation, in this case, they will lead to the aberrant demethylation patterns. In this case, as we will cover later in this lecture, that to maintain such Chromatin structure, there's a protein called CTCF need to bind to the DNA and some sort of like seal the Chromatin domain. But once you have the IDH mutation, the DNA binding motif, CTCF motif, is methylated. So as a result, the protein can no longer bind to their motif. As a result, the 3D structure is disrupted. And then the enhancers can now talk or contact the oncogenes and further drive its expression up by almost 4-fold. As a result, it contributes to cancerogenesis. So here is another example. In this case, this is actually a SV or structural variant lead to the change of 3D genome structure. Okay, so in the normal Chromatin 3 in the human blood, right, there's an enhancer regulating GATA2 through Chromatin looping events. So GATA2 is expressed, and this oncogene EVI1 is silenced, which is good. But a main category for AML leukemia patients, there's a inversion 3. Is this big chunk is inverted, but no DNA sequence or mutation happened in the enhancer. But because there's an inversion happened, so this enhancer now is shuffled pretty close to the EVI1 and form Chromatin looping events with the EVI1 and for the drive up expression for this oncogene expression and for the contribute to leukemogenesis. So I want to say that this is actually the so-called enhancer hijacking, and the enhancer hijacking concept began to gain popularity only in the past few years. And I think this will be a really, really exciting research directions for the cancer studies.